Okay, well, welcome everybody. Welcome to track 3, where we'll be exploring pest management. My name is Hunter Blanchard, and I serve on the annual conference planning committee, and I will be your host for this session. We encourage you to please join us with your camera on if you're able to. We're sure that you're going to enjoy hearing from our next presenter, Nick Brazee, who will be presenting on beech leaf disease in Massachusetts, from bad to worse. I'd like to take a second to thank our sponsors for this year's conference, Kelly D. Norris, who many of you saw speak yesterday, and Ernst Conservation Seeds, who we use for a lot of projects at the garden. I have a few housekeeping notes to share before we get going. Feel free to post your questions for the speaker in the Zoom chat. We'll do everything that we can to have all questions answered in this session. And if they're not answered, you're invited to go to Track 3 Speaker Garden after the session for further opportunities to explore your questions more fully with our speaker and others. We want to thank all of you who are ELA members. So all the ELA members in this room, please raise your hand or add a reaction to your Zoom screen if you are currently a member. We'd love to see your visibility. Your membership helps us shape the direction of the organization, and we are so grateful for your contributions. Thank you for offering feedback and suggestions about programming, for writing articles for our publications, for volunteering your time on working committees, for hosting eco tours and regional meetups, for presenting webinars, and for helping ELA work towards our vision that all people understand and respect their connection to the natural systems and take informed action to support ecological landscapes. Your membership comes with loads of benefits, including discounts to great events like these and access to our members-only quarterly publication, Into the Weeds, and other online member-only events. You can go to ecolandscaping.org/membership for more information. So thank you for your attention, and now let's get started with the session. Perfect. Nicholas Brazee is an extension plant pathologist at the University of Massachusetts. He directs the UMass Plant Diagnostic Lab, providing woody plant disease diagnostics and management support for green industry professionals. In addition, he conducts research on fungal pathogens of urban and landscape trees. Dr. Brazee has a PhD in forest pathology from the University of Massachusetts. Please join me in welcoming Nick Brazee.
All right, thank you, Hunter. Thanks everyone for your attendance today. So I assume the sound screen looks good. Everything's okay there. All right, excellent. Okay, so we are going to discuss this cheerful topic on this beautiful late winter day here in southern New England. Oh, actually, it's the opposite of that. It's beech leaf disease from bad to worse, and we really haven't hit the bottom yet. So it's only going to get worse and worse from this point on. But we will document it and take it all in as it occurs. So this is my first time speaking about beech leaf disease solely in a talk. So I thought it was worthwhile to take a step back and look at beech and look at some of the factors that have really led to this unfortunate outbreak of beech leaf disease. So if we look at American beech, Fagus grandifolia, it's the only species in the genus in North America. Later on, I'll discuss some of the diversity in the genus outside of North America, but it's our only species. As you can see from the map here, shaded in green, has a pretty significant range throughout Eastern North America, certainly all throughout New England, the Northeast, but it really extends much deeper down into the South than I think a lot of us recognize, into parts of East Texas and throughout Alabama, parts of Mississippi. So it has a pretty significant range. Obviously, as it gets down into the South, it's a little more sparse and on wet mesic sites. It is extremely shade tolerant. It's one of, if not the most shade tolerant deciduous hardwood in North America. So extremely low light levels are tolerated by American beech, and that has helped to unfortunately create the conditions that are so conducive for outbreak. It is a slow growing tree. It can achieve some pretty significant ages of 300 to 400 years old. The range is going to be important going forward in the future when we think about preservation and reintroduction. So when seed is collected from potentially resistant trees, it's going to be important to document where that's from. If it's from, northwestern Florida, it's probably not very well adapted to a cold climate on the northern extent of the range. So there's going to be a lot of examination of where resistant trees are for reintroduction. So that's where the range is important. I wanted to just highlight kind of the core habitat where we find American beech in New England, and that's the pure northern hardwood forest. So if you're familiar with forest types, the northern hardwoods are primarily composed of beech, birch, and maple, and there's a lot of associates, and we'll talk about that in terms of what the future looks like in these forests. But the core northern hardwood forest is beech, birch, maple, and you can see that distribution there highlighted in green. So if we look at Massachusetts, it's primarily on the Berkshire Plateau and extreme western Mass. And then as we extend up throughout Vermont, parts of northern New Hampshire, lots of Maine, the Adirondacks, the Catskills. But if we expand this out to also include hemlock in this forest type classification, we get a much broader area that is dominated by forests with significant amounts of beech. And unfortunately, it's truncated as we get into Canada and parts of the Midwest. But we're looking at this area in the Northeast of roughly 34 million acres that is composed of forests where beech dominates in some capacity. And even as we get, I've been surprised even in areas of Eastern Mass, some of the islands, the North Shore, Southern Connecticut, Rhode Island, there are some pretty significant pockets of beech, areas I wouldn't necessarily think would have beech where we see it. So all throughout New England, beech is a very significant tree species. And to highlight that further, we will look at some U.S. Forest Service forest inventory and analysis data. So this is a photograph that I took many years ago out in the Berkshires, and it really encapsulates what a lot of our northern hardwood forests look like today. We have a couple of overstory sugar maples. There's one on the right, one on the left. There's a yellow birch kind of lingering in the background towards the left side. But the composition of this forest is just completely dominated by beech. There's a few beech in the overstory. There's not many left. We'll talk about why that is. But the understory is just dominated by American beech. So, we look at the composition of beech throughout a lot of our forests in New England. And, there's areas where 50, 60, 70% of the stand could be composed of American beech. If we look at the FIA data, the Forest Inventory and Analysis, and you may think, well, we don't have a lot of Forest Service land here in Massachusetts, or maybe we don't have any at all. The Forest Service has permanent plots in all 50 states, and every 5 years they do an inventory to look at composition and structure and a lot of other characteristics. So despite not having a lot of federal land, we are part of this inventory system. And the data sets are rich and really fascinating if you dig into them. Now, in Massachusetts, on our forest lands, of all trees that are 1 inch or greater in diameter, we have an estimated 1.44 billion trees, which is pretty staggering for a relatively small state like ours. It really shows just how many trees there are in this region. Of that roughly 1.5 billion trees, over 10% of that are American beech. So it has this really outsized importance in Massachusetts and in a lot of surrounding states. Hemlock, white pine, red maple, those are the top 3, and beech is number 4, but over 10% of our forests are composed of American beech. What's really interesting, if we go further, is 87% of that total, over 130 million, are in this 1 to 5 inch diameter class. So it just kind of goes back to that photo where the vast majority of these beech are small sapling to pole size trees in the understory. So it dominates the forests, but mostly as an understory tree. There's very few overstory beech trees left. Beech is really significant in the core northern hardwood forest because of the mast that it produces. So there's a large variety of wildlife, birds, mice, squirrels, chipmunks, bear, deer, foxes, grouse, ducks, blue jays that really utilize beech nuts. And if we look at the core northern hardwood type, the beech, birch, maple, it's the only nut producer in that type. Now, when we get down here in Massachusetts, we get northern red oak mixed into a lot of our northern hardwood forests. So there is additional mass there from the acorns. But as you get up into northern New England and you see oak drop out completely, you know, beech is the only game in town for any kind of mast. So it really is important ecologically from that perspective. And beech leaf disease is really spawning a lot of focus and attention on just what specific organisms are very dependent on beech as, you know, as we lose it from the ecosystem, what kind of cascading effects that will have. Okay, so in order to properly talk about beech leaf disease, we have to step back and we have to recognize the devastation that beech bark disease has caused. So a similar acronym, BBD instead of BLD. What we're looking at here, this is a mature beech, a photograph I took a few years ago in early October of the ravages of beech bark disease. This is the fungus Neonectria, and these little red masses that are rupturing through the bark. These are fruiting bodies, so it goes through its sexual reproduction in the fall. Very conspicuous, very distinctive. We can see this tree is deep in the throes of infection at this time. And we have had beech bark disease in New England for well over a century now, so it is a naturalized disease complex, and it has really shaped the northern hardwood forests as we see it today and made conditions, unfortunately, more conducive for this beech leaf disease outbreak. So beech bark disease, if you're not super familiar with it, it is a pest pathogen complex. So what happens is there are two beech scales, Cryptococcus vagisuja and Xylococcus. They will infest the trees, and they feed on the main trunk and large scaffold limbs, and they use, like many scales, they have a piercing, sucking mouth part, and they will extract fluid from underneath the bark. And beech have very thin bark, and especially when you get into the valleys of the ridges, you know, they don't need a particularly long mouth part to get down into the sap. And the feeding that they, the feeding injury that they cause create these little wounds that are then susceptible to Neonectria, this fungal pathogen. And there's two species of Neonectria, Ditissima and vaginata, that will subsequently move in and infect the trees and cause beech bark disease. But it's really, if we didn't have the scales, if that wasn't an issue, we would have some isolated cankering from Neonectria because Ditissima is a native pathogen here, but we wouldn't see the scale of destruction. So the scale is important, and it's often overshadowed because of the damage that Neonectria causes. So both are required in order to cause beech bark disease. So as I mentioned, Neonectria ditissima is a native pathogen, and perhaps in the forest you've seen this target canker of birch, on yellow birch and black birch in particular. It's very common throughout our forests where you get this kind of slowly expanding canker where the fungus will grow out and break through the wound wood, and then during the growing season, the tree will form a new layer of wound wood. And so you get this kind of back and forth between the pathogen and host, and it creates this kind of target appearance over time, which is why it's called target canker. You can see just from this image how different the symptoms are from birch to beech. We see a completely different array of symptoms, and we can find Neonectria we can find it on basswood, on tilia. I get other samples in the diagnostic lab, hornbeam, other trees like that are suffering from Neonectria canker. So it really highlights how the scale is so necessary to make the tree susceptible for Neonectria to really operate in a manner in which it normally does not. So this image on the left here, this is a badly diseased tree. So as Neonectria establishes and causes disease, we get cracking, splitting, sloughing of the bark. It's important to note that a fungus like Neonectria it's not causing decay of the wood. So it is after sugars that are in the phloem and the outer xylem. So it's attacking the bark tissue to get down to the phloem to those sugars, but it doesn't cause wood decay. So it's not attacking the the structural support of the tree. It's not getting into that old xylem tissue and breaking that down. But as the bark is killed, you get this very rough, cracked appearance. One thing about beech bark disease is it does tend to move pretty slowly over time. It takes many, many years for a tree to die from beech bark disease, and trees can live on in some state of decline for a long time with beech bark disease because it's so slow when it does establish on a tree. And so unfortunately, as these wounds develop, the trees then become susceptible to wood rotting fungi, so fungi that can actually break down wood tissue. And what we're looking at here, this hoof-shaped conch, this is Phalinus ulni. Phalinus is very common throughout the northern hardwoods on beech, birch, maple, and it is a common trunk rot pathogen throughout a variety of forest types. There's a range of different species, and so it will gladly take advantage of a stressed and damaged beech. There's a multitude of infection sites as a result of beech bark disease. And so you get these secondary organisms that are able to invade, and then the trees often they'll die standing or they'll suffer stem failure. And so this pattern of decline and death, where you have these kind of small gaps that are forming in the forest, it really, this is the typical disturbance regime here in the Northeast, where we have wood rotting fungi and we have windstorms, and you get this small gap forming, or one to a few trees that develop, as opposed to a huge wildland fire that sweeps through and kills a large number of trees, or some mass event where a large number of trees are dying out. As a result of this kind of small gap phase disturbance regime, there's not a lot of resulting light that reaches the forest floor. So you can see how in a climax northern hardwood system, you're really favoring basically sugar maple and beech, the two most shade tolerant species. So single tree mortality, you're not getting enough light on the forest floor that's going to allow red oak or another tree that requires a lot of sunlight to really establish. So the aftermath of beech bark disease is we have this loss of mature overstory American beech. Their numbers are dramatically reduced from the time period prior to the introduction of the scale and the fungus. But surprisingly and really interestingly, when we look at these individual tree species epidemics, instead of seeing an overall decrease in beech numbers, we actually see an increase. And that is because beech does very well from these root sprouts. And so as the trees die back, you get a number of sprouts from the root system, and we develop what are known as these sprout thickets. And this is why we just have so much beech in the understory of a lot of these forests. As these overstory trees died back, these sprout thickets formed. You just have dense shade where a lot of other trees can't establish, and it's just kind of this feedback loop where we continue to see a lot of beech establishing. There's other factors as well that I'll talk about later on. But as I said, the percent stand composition in a lot of these forests is just dominated by beech, even though there's relatively few of them in the overstory as large mature trees. The smaller diameter trees, so you have a beech that develops from a sprout, it grows, it can remain free of the scale for a number of years. The diameter is just so small as the scales are kind of blowing around. There just isn't a lot of surface area for the scales to land on. So, if you go to these northern hardwood forests and look at a lot of these young beech, they're relatively healthy until they get to a certain diameter, and then they start to accumulate the scale. It builds up kind of slowly over time, and then those feeding wounds accumulate, and then the fungus can invade. So it is a relatively slow process. So we have this image here, this picture I took. We have a badly diseased tree on the left, and then a resistant tree on the right. So there are some American beech that are resistant to beech bark disease, but their numbers are just unfortunately so few that we don't see any significant numbers of resistant beech out there. They're pretty hard to find. You can find in little pockets here and there, but there is no major resistance. And that is very typical of an introduced disease complex, in this case, a pest pathogen complex, where we have an insect and fungal pathogen. Even though we have a native fungus, Neonectria ditissima, we have a non-native scale that has really turned the tables on beech. Okay, so this kind of sets the stage for beech leaf disease. So we have this huge population of beech kind of as an understory tree, and then beech leaf disease enters the scene. And the acronym BLD is very easy to confuse with BBD. So beech leaf disease is caused by Liddylincus crenatiae subspecies McCannii. This is a nematode, and you see this increasingly just abbreviated as LCM. So if you see LCM, that's the abbreviation for Liddylincus. On the left, we have symptoms on an American beech, and then on the right, a European beech, Fagus sylvatica, which is very common in our managed landscapes. So you can see the symptoms can vary a little bit, but overall they're pretty consistent, and I'll go through the symptoms of the full range of symptoms that we see on trees. It is unprecedented throughout the history of plant pathologies, through our time period of studying pests and pathogens, to find a nematode that can cause a widespread mortality of a large forest tree. It just doesn't happen. Now, nematodes are abundant in all kinds of different environments, in aquatic environments, in soil. They're a big problem in managed turf grasses. They're major vegetable pathogens. They are these microscopic, non-segmented roundworms. They're actually part of the animal kingdom. They're abundant in aquatic organisms. If you eat fish, then you're eating nematodes. They're pretty common in a lot of other mammals. So they're just kind of everywhere. We could go out and take some soil, and right outside wherever you are, and there would be nematodes in the soil. They're just one of the most abundant free-living organisms in the world. But there's never been a case where they have infested a large tree. There are some cases of nematodes causing problems on forest trees, the pine wilt nematode, which is a problem on Scots pine, but never something on this level of a tree like American beech suffering outright mortality from infestation by a nematode. What's interesting about nematodes is you think, okay, it's an animal, but it's very insect-like. I think we would all agree. You think that would fall under the realm of entomology, but since the beginning of plant pathology in the 1850s, nematodes have been studied by plant pathologists, and it's a further subdivision within already a niche profession. So nematologists are typically housed in plant pathology departments, and increasingly there are fewer and fewer nematologists out there. So perhaps beech leaf disease will spawn a new generation of nematologists, but it's getting harder and harder to find these individuals at universities and government research labs. This is the current distribution of beech leaf disease. You can see I tried to choose a color here on the left, this light orange star to indicate the epicenter, the origin of beech leaf disease in Northeast Ohio. It was first detected in 2012, and I'll talk about the distribution in Ohio a little bit later on. It's moved relatively slowly through the forests of Northeast Ohio. It was spreading kind of slowly initially. It was kind of radiating out from the center into Western Pennsylvania, extreme Western New York, up into Canada. And it was on our radar. We started to really get concerned about it in 2018, I think. And it took a long time for researchers to identify that the nematode was the causal agent, because there was no precedent. They were finding nematodes, but thinking there must be something else involved here, like a bacterium or a virus or maybe a fungus. And there are these microbial symbionts that are associated with nematodes. So if you look at nematodes in roots or soil, they often have bacteria that they're associated with. But really, it took a while for researchers to say the primary cause of the symptoms are the result of the nematode feeding by itself. And that was a little bit controversial at the time, but now is really ironclad, is irrefutable at this point, that the nematode is primarily responsible. Unfortunately for us, if you look down into southwestern Connecticut and Long Island in 2019, that's when it made its first appearance. And that was a significant jump at the time. There was a large geographic area between the most recent the most recent finding in kind of central Pennsylvania and New England. So we thought we had some time before it was going to kind of radiate out and get into our region. So it led to just a lot of discussion about what the potential vectors are. And I don't talk about that a lot here in favor of other subjects, but birds were suspected as being a primary vector at the time. We now know that there's, various insects, probably any kind of mammal that's in a forest system, but rodents like chipmunks and squirrels can be vectoring them on a small scale. But really, wind and rain is important for local dispersal. I will mention that a little bit later on. But first found in Massachusetts in 2020 in Plymouth, and it has since spread just like wildfire throughout most of New England. We'll see it spread into northern Vermont, northern New Hampshire, the Adirondacks, unfortunately, in the coming years. And so it's been established for several years now. And so we're seeing trees that have suffered multi-year infestations as it builds up. And so as we get into 2025 and 2026, I think we're going to start really to see the mortality start to develop from beech leaf disease, and especially in the eastern part of the state where it was first introduced. Okay, as I said, we'll go through the hosts and susceptibility here of what we know. We know American beech is highly susceptible. It has no resistance. There may be a few resistant trees out there that we can identify, but for the most part, it is highly susceptible. European beech, unfortunately, is also very susceptible. Oriental beech, you may have heard of. It's not widely known or planted as an ornamental. The Arnold Arboretum does have a few. It is native to kind of Eastern Europe and Western parts of Asia, but it's considered a European species. We believe that's also equally as susceptible as Fagus sylvatica, so it seems to be right on the list of susceptible trees. When we get into Asia, that's where the diversity in genus Fagus really increases. And we know that Japanese beech, Fagus crenata, is resistant. So there has been a recent team of Forest Service professionals and some folks from the Agricultural Research Service, ARS, that recently went to Japan in search of the beech leaf disease origins. And it is now, there's a lot of evidence to suggest that it is native to Japan. And Fagus crenata can be infested, but exhibits a high degree of resistance, suffering only very minor leaf loss as a result of infestation by the nematode. But there's several other species that are in China and other parts of Asia, Fagus japonica. And again, there's some crenata at the Arnold Arboretum, and they are diseased there, but I believe they have some japonica as well. I need to confirm that. But some of these other species on the bottom, like sinensis and hayate, if I'm pronouncing that even remotely correct, these are beech I've never even heard of before. I mean, these are so far outside of even, kind of niche ornamental trade, things that we've just never seen. So there's still work to do on which of these are resistant and which might be susceptible. But for us, since grandifolia is the only game in town, we know it is highly susceptible to beech leaf disease. Okay, symptoms. We see the interveinal banding. So banding in this convex cupping that develops. We can see here on this American beech, we see distortion and curling of the leaves as they become infested with the nematode. We can see really high levels of stunting and distortion. This was a very prominent symptom in 2024 on infested trees, where to me, this looks like a group 4 broadleaf herbicide injury, where this really high level of distortion that is outside of things that we normally see on disease and insects. It looks like a chemical injury, but it is in fact caused by the nematode. And then this, at times, the second flush of foliage, which prior to beech leaf disease, I didn't even know beech could do this. Now you see trees with determinate growth rates like oak, they'll have a second flush at times if it's wet. But I wasn't even aware that beech could do this. It's highly stressful for the trees. They have to exhaust their starch reserves to kind of push this second set of leaves. So it usually is an indication that a tree is under some really serious stress when they do that. Some additional symptoms, this image on the left, you can have leaves that are irregularly thickened, so they have this kind of leathery appearance and texture to them. And then we get this early senescence within the bands. So the nematodes are kind of clustered in these bands here, and there is an increasing carotenoids and other compounds, and this may result in some early senescence within this area. So there's been some speculation that this thickening and dark coloration in the bands is potentially a means of protection for the nematode. They're stimulating this production to provide them some protection from ultraviolet light. So there's still a lot to work out with features of the nematode, what it's doing, but we know that it's highly destructive and capable of causing mortality. So let's go through the life cycle here just to illustrate how this nematode is causing injury. So in the spring, the leaves are emerging fully symptomatic, and the nematode populations are generally low at that time. So we're getting, and I'll pause on that statement until we get later on here. During the summer and autumn period, that's where we start to see the populations of the nematode increase. So eggs within the leaf are hatching, the nematodes are increasing in abundance, and then they are migrating from the leaves. So they're exiting the leaves through the stomata, and then they are crawling across the surface of the twigs, or they're being splashed and blown to nearby trees, and they are looking for the buds for next year's growth. So they're looking to get up to the bud and make their way underneath the bud scales and get into those buds. And the reason is that's where they will feed and cause their damage. So they're going to overwinter inside the bud. And so really, another pathologist said a few years ago when this first started that this really should be called beech bud disease. And it's a good thing that it isn't, because it would have the same acronym as beech bark disease. But this is where the injury is taking place. So the nematodes are inside the bud, they're feeding on the leaf primordia. And that's why when the leaves elongate and emerge, they're fully symptomatic at leaf out, is because that damage has taken place inside the bud. So if we were to go out right now, late winter, collect buds on diseased trees and dissect those buds, we will see those damaged leaf primordia inside there, and we'll find nematodes that are present within the buds. So when nematode populations are low, the symptoms will be less severe. So we'll see this banding that we see in this upper right-hand image. We'll see the banding and some of the convex cupping and maybe a little bit of distortion, but overall we'll see light symptoms. But if there's a significant population in the bud, and we don't have a threshold value yet, that's one thing that will be interesting to find out, how many nematodes it takes. But if there's a lot of nematodes and there's very destructive feeding, the bud is just killed completely. The bud's aborted and it's not producing any new growth. And that is really, that right there is how this disease is so devastating. When the buds are overwhelmed with nematodes and they die, it can't produce any new woody tissue and no new foliage. And so that's where we start to see dieback and thinning throughout the canopy. And then it just exhausts the tree's natural starch reserves, and it just basically kills the tree from lack of new growth. This was a really interesting study that came out last fall, deciphering the vectors. As I mentioned, there's been a lot of speculation and a lot of interest in understanding how the nematode is spreading, because it's this tiny microscopic roundworm. How is it moving so efficiently and so quickly? And basically, what we know now is that there are just these astronomical populations of the nematode out there throughout our region, and basically throughout every area where beech leaf disease is established. And one of the primary ways that they're spreading locally is just through wind and rain. And so this figure here from the study, I just want to go through it quickly here because it's just really fascinating. So on the y-axis here, we have the mean number of nematodes recovered. So basically what these researchers did, they went to a forest that was infested with beech leaf disease, and they set up these funnel traps at various distances underneath these trees just to see how many nematodes they could collect. And it's a collection protocol that's often used for if you're looking to get nematodes out of soil or out of turf or some other medium, using these funnel traps. On the x-axis here, we have a time period from September 9 on the left to November 8th on the right. And the autumn period, as I said, that's the pulse period of movement for the nematodes. So they start to move in midsummer, but the numbers really ramp up in autumn. That's when the largest numbers of nematodes are on the move. And so when the collections of nematodes per day are correlated with some weather data, we see that it takes only minor rain events to cause these kind of peaks in recovery. So these blue bars here are spikes in nematode recovery. And if we look at the numbers here, the top figure, miles per hour, that's the wind speed. Then the second figure is the amount of precipitation, and then the relative humidity. So you can see going through these, 2.65 mile an hour wind, very light, 0.06 inches, and 82% humidity. And we go through all of the pulse periods of dispersal. The largest amount of rainfall was 1/4 inch. In some cases, it was 0.005, so half of 1/100th of an inch. So really any kind of minor wetting event, any very light rain and light wind is enough to move these. So we think if we have more significant precipitation with stronger winds, that would blow them around much further. So they exit the leaves, they're migrating on the stems towards the buds, and then they're getting splashed and blown around. So some of you may be thinking, well, it was extremely dry last fall around southern New England, basically barely rained from August 20th to November 20th. But going back, even these minor wetting events are enough. Here in the Connecticut Valley, late summer, early fall, we get some really dense fog where it's so dense that you get precipitation, you get moisture dripping off the trees in the morning. So I think just having high relative humidity in the morning is enough wetting for these nematodes to disperse. And unfortunately, what's very unfortunate is they can survive periods of desiccation where they can dry out completely on the twigs, or they come completely desiccated, and then you get this minor wetting event and they're rehydrated and they can move on. It's almost like they're robotic in that sense. And if you've heard Matt Borden from Bartlett Tree, Bartlett's done a lot of the work on the management side that we'll talk about, but they were talking about, he had nematodes that were in the freezer for like a year. They were collected buds, they were live nematodes, they froze them for a year, pulled them out of the freezer, thawed them out, and they wake up and start moving again. So they're incredibly resilient and resistant. So that is unfortunate. Another interesting study that came out recently looking at the anatomical and physiological consequences on American beech. So foliage that is infested by Liddylingus, it exhibits lower photosynthetic capacity, so it's not photosynthesizing as well. It has increased rates of respiration. Respiration is the primary means of cooling, where water is exiting the stomata, it's capturing heat on the surface and evaporating, so this evaporative cooling. And when it's hot and dry, like it was last summer, there's a lot of respiration. So they're photosynthesizing less, they are using more water than they would normally, and they have these increased leaf construction costs. So just the resources required to create a leaf, because you have this unnaturally thickened tissue. And in diseased areas of foliage, in some cases, we're 250% thicker compared to healthy areas. So that's just exhausting resources that the tree has just to produce foliage. So it just kind of browbeats these trees until they become unfortunately attacked by other insects and disease. Okay, when we think about management, what are we going to do? How do we slow this down on a few select trees, or just where are we in the trajectory of the disease wave that's moving through? So right now we're somewhere on this exponential growth curve. When we look at the population of the nematode, it is increasing rapidly, exponentially throughout the region. At some point, it will reach a peak, and that's because beech has started to die off in large numbers. And so this pattern is pretty typical of an introduced pest or disease and a collapse of its host. And beech mortality will start slowly as it is now, and then at some point it's going to crash down, and hopefully it results in some kind of carrying capacity, so to speak, where the population of the nematode will crash and drop down to some new normal, some naturalized level as the population of beech declines. And hopefully we'll still have some beech left. We'll have beech out there in our forest and our managed landscapes, and we'll have a much smaller population of the nematode to deal with, and then we'll have more management options. But right now we have this ever-growing population of the nematode, and likely hasn't peaked in a lot of areas. So we have to let it peak and then decline as beech dies off en masse. Okay, so from the early stages of the disease, as I said, it was first detected in Ohio, so a lot of the work has happened there initially. What we were hearing was that the phosphites could provide some level of control. And if you're not familiar with phosphites, these are mono and dipotassium salts of phosphorus acid. And this product has two modes of action. It is weakly fungicidal, and it also works to stimulate the natural defense response of the tree. And so you may be thinking, oh, he said fungicidal, this is a nematode. How does a fungicide work against a nematode? Well, it doesn't. So there have been some very well done artful studies showing how phosphites in improving the defense response of the tree actually allows the tree to overcome a pathogen. And one of those in particular is Phytophthora canker on beech. So this initially was met with a lot of positive views from arborists, because they're saying, I'm already treating beech with phosphites for Phytophthora canker, so this should provide some resistance and provides me a good means of control going forward. The phosphites can be applied very locally to the soil around the root flare and then are taken up. They're highly mobile in the xylem and the phloem. They can also be applied as a lower trunk spray, and they penetrate through the bark with a carrier, and then they're taken up into the canopy. So this was good news. What we have learned in subsequent years is that the phosphites alone seem to do nothing here in the Northeast to control beech leaf disease. Davy Tree continues to report that they're seeing good control in Ohio, and I believe them. They have good data, but there seems to be a disconnect in the severity between what we're seeing here in the Northeast and what they're seeing in parts of the Midwest. So there's a little bit of a difference there. Phosphites are very broad spectrum, so they have utility in many cases against an array of different diseases. So as I mentioned, when these trees are stressed, they start circling the drain and other organisms move in on them. So the phosphites can have utility against cankering fungi or other diseases that would move in on a stressed tree. And because they're so easy to apply, they can have some utility. But for beech leaf disease alone, the phosphites don't seem to be doing much. Okay, going back to the map here. So if we look at Northeast Ohio, this is where it was first detected. If we look at the distribution, it took until 2023 until beech leaf disease had spread throughout Northeast Ohio. And you might think, well, Ohio, how much beech do they really have? Well, I went to the forest inventory analysis data. They have about 150 million American beech, the same number as we have here. It represents a smaller, much smaller percentage of their overall trees, about 4% of their trees, but that's still a lot of beech. And you might think, okay, there's still a lot of the state left, but you get into the western and southern parts of the state and forests really drop out. But it seems to be spreading much more slowly there. And maybe that's because beech isn't in these higher percentages and concentrations within the stands like they are here. But Davy continues to report that they're seeing good control with the phosphites. So it's something that we'll look at further. There's another chemical that's available, another product called Broadform. It is a combination product that utilizes fluopyram and trifloxystrobin. Fluopyram is a relatively new fungicide and nematicide that has been used in turf grass, vegetables, and ornamentals. It was a relatively unknown product until it was discovered that it had utility against beech leaf disease, and then it started flying off the shelf and you couldn't find it anywhere until the supply increased. It will kill nematodes inside the leaves before they can migrate to the buds. And so treatments with Broadform, even a single application of Broadform as a canopy spray, have shown to provide some level of control, in some cases very good control. So Broadform right now represents the primary means of control in our managed landscapes for knocking back the population of the nematode. It may not eradicate it, but it can significantly reduce the nematode. And I've seen trees that have been treated from anywhere from May into September. And so there's been a lot of debate and discussion and research on when the exact timing should take place, but it seems to be there's a wide window. So initially there was a supplemental label that came out, which is called a 2EE recommendation to allow Broadform to be used against beech leaf disease because it was not on the label. This has been modified. Initially, it was 8 ounces of Broadform per 100 gallons of water that has since been dialed back, and the research has shown that much lower concentrations, maybe even 1 ounce or less, is sufficient. And I think it's very much in our favor to use less of this material as possible, which I'll talk about in the next slide. And the initial application interval was 7 to 14 days. That's been increased to 21 days. And so Bartlett, again, they've been doing a lot of work on this front. They tried to get this minimum concentration down even lower, but met some resistance from the state. And so they're working to continue to reduce that amount, to limit the amount of broad form that is applied. And the reason being, this is an interesting article that I was reading this past fall. Just, many of you are certainly aware of PFAS chemicals and well water and soil. And when I was at the University of Washington in the mid-2000s, a big topic of research at the time was using these biosludge materials, this waste product from paper mills, and applying it to forests that had been, so these Doug fir forests in Washington and Oregon, they would clear cut them. And then the feeling was, let's put this biosludge onto them, this waste product, and this would be a win-win. We could utilize this waste product and it would be a fertilizer for the regenerating trees. And, now we know that these sludge materials from paper factories and a variety of other industrial sources, they're just loaded with PFAS chemicals, gets in the soil, gets in the water, subsequently is in wildlife. And so they're just terrible. And there's been increasing concern and attention about PFAS materials that are in pesticides. And so currently there's 66 PFAS active ingredients registered in the United States. So this process of what constitutes a PFAS molecule. So these molecules they have a chain of linked carbon and fluorine atoms. And this CF bond is extremely stable and breaks down only very slowly over time. This process of fluorination to create these molecules is relatively new. This is a newer chemistry. And so many of these PFAS chemicals that we see have been produced, in the last 20, 30 years. There's not a lot of avenues for creating new pesticides. So this fluorination process has resulted in a number of new chemicals. So of these 66, like half of them have come out over the past, say, 30 years. Unfortunately, including fluopyram and trifloxystrobin, the two active ingredients in broad form. So this is a big concern going forward, where we want to treat for beech leaf disease and save, in particular trees that have significant cultural and historical value, but we don't want to subsequently make the problem worse. And this is where I've, in speaking with arborists, which is my primary audience and group, we talk about the fact that deciding not to treat for beech leaf disease is a management decision. That is, an acceptance that this is a terrible disease. It's sweeping through. It's likely to kill the tree, and adding, pesticides may only make the problem worse. The worst case scenario is you've got beech leaf disease, you treat your tree, and it still dies. So then you've added pesticides to the environment, and the tree ultimately has died. So the other downside here is that fluopyram is really what we want to use in controlling the nematode. The trifloxystrobin is like a throwaway. It's just part of this product. There isn't a single product. There isn't a fluopyram only material that we can use for beech leaf disease. This is the only one that's available right now. And so the trifloxystrobin has no utility whatsoever. And so it's just getting applied in mass without any need. And so that's obviously not part of a good IPM program. 2024, there was a special local needs label that was approved by the state for the use of ArborTect. So this is a fungicide and a nematicide that is injected into the root flare. So this is widely used for Dutch elm disease control. So maybe you've heard about it for protecting historical elms. It's injected into the root flare. It's taken up into the xylem and provides some control. We don't have as much data on the ArborTect as we do the broad form, but some of the early trials suggest that it is working quite well in controlling the nematode. And so this would be vastly preferable as opposed to broadcast spray of the canopy where there's drift. We can just target the injection into the root flare and it stays within that individual tree. There is a phytotoxicity risk, so it has to be applied once the trees are fully leafed out. The concern is that, this injection site wounding on the root flare, how are beech going to tolerate that? So, right now, the only information we have is from American elm, which is so tough and resilient that you can do almost anything you want to an elm and it will continue to persist. And I've worked with elms where they've been under these injection cycles for 20 or 30 years of getting an injection every 2 to 3 years. And that you can't reuse the same injection site, so you have to drill a new hole. So the trees are just loaded with injection site wounds around the base. And elms tolerate that very well. But how are beech going to tolerate that? If we think about 10 years out, 5 injection cycles, you know, all these injection site wounds, what's that going to mean for tree health? But honestly, at this point, if we're injecting for beech leaf disease, it seems relevant to only look maybe 1 or 2 injection cycles out, because we're at this point of peak disease severity. So we really can't look that far down the road. If we're looking to save a tree, we can only maybe look in the next few years. Here's a tree on the UMass campus that also got this Moget injection with Tebuject. Unfortunately, right now, trees are getting this kind of kitchen sink approach where they're getting phosphites, they're getting broad form, they may be getting other chemicals as well. So it's hard to tease out how the individual treatments are working by themselves because multiple treatments are happening. This is a tree that in 2023 had very light severity. It's an important tree. It's one that the campus arborists really want to protect and save, a mature European beech. Disease severity went from very low, minimal in 2023 to really severe in 2024, made just a rapid increase in 1 year. 2024, second flush of leaves, high distortion, banding, undersized foliage, very thin canopy. So it really illustrated how quickly disease severity can ramp up. So right now, everything is being done to protect this tree, hopefully to get through this initial wave of the disease. And then as the population reduces, the treatments could back off and we'll know more about how to make less intensive treatments. There is another product out there called Biosar, which is the active ingredient is chitosan, which is a natural polymer that is non-toxic and biodegradable. It has been widely tested against a variety of soil nematodes that are in the soil that attack roots, and it appears to work quite well in many of those cases. Unfortunately, the way it's labeled, right on the label, controls beech leaf disease. Okay, if you're going to say that, I would like to see some efficacy data, some trials. It is registered under this exemption, 25B, that exempts it from EPA regulations on efficacy and toxicity. So we know what are, the toxicity of this material, and we don't really know much about the efficacy. It's sparsely used. To the trials that I've heard, just anecdotally, it doesn't seem to be working. I've seen many studies where it works well on soil-borne nematodes in the soil and roots, but this nematode is in the foliage and the above ground parts of the plant. So that's the total opposite environment and medium. So it seemed to me to be a bit bold to put controls beech leaf disease right on the label without a lot of data to back that up. And unfortunately, what I'm hearing is that it isn't doing a whole lot, which is unfortunate because it would be a great option going forward, being this chitosan, this natural product. Okay, so right now, what represents good control? We don't need perfect, healthy trees with no symptoms at all. We could have abundant leaf banding, but an otherwise full canopy and foliage with a normal shape and size. That represents good control going forward right now, where we are in the beech leaf disease epidemic. When we have these undersized, distorted leaves with a thin canopy and a second flush of foliage, if treatments are taking place, they're not working. And really, there should be a hard look at whether or not they continue. For trees that are untreated, this is what we see after multiple years. We see the intensity increase as the nematode population continues to expand and develop. And initially, folks in Ohio were saying it would take about 10 years to kill a tree. But in southern Connecticut, where it was first established, Rhode Island, in some cases, 2 or 3 years of infestation is enough to cause mortality. So we're going to start to see that wave of mortality build now that it's been established for a few years in the state. So some questions for this year, things we want to know that nematologists are working on. How many nematodes per bud to cause death? It would be nice to have a threshold value so that when we count nematodes in the buds, if we can kind of get a rough estimate, that it takes 1,000, if it takes, 600, just to have some threshold to know how severe the disease is going to be on that particular tree going forward, and what kind of management needs to take place in order to bring that number down. The refinement and timing and the reduction in rates of broad form. especially now that we know that it is technically, you know, and there's variation in how serious some of these PFAS chemicals are. And another aspect of that study I forgot to mention is, you know, there can be PFAS. There's PFAS in the plastic bottle that gets leached into the product. So even something like insecticidal soap could have PFAS in that material because it's leaching out of the bottle. So there's a lot of ways that these PFAS are getting into chemicals that we may be using, even if it's not the active ingredient. So these things are just so pervasive throughout our lives. And you know, if you were at the talk yesterday about the plastic pots, you know, it's just kind of hard to get away from plastic in general, just kind of taking over our entire lives. One thing that I'm going to be doing across campus is collecting leaves on some of our beech and looking at the foliar nutrients. And we know that beech leaf disease is throwing off some of these values. And so I just wanted to highlight that there's a number of extension labs that do these nutrient analysis. It was a service that was discontinued by the UMass Soil and Plant Nutrient Testing Lab. That's separate from where I am in the Plant Diagnostic Lab. Went on hiatus during COVID, but they brought it back. And so I think if you're working with beech and trying to save them, a foliar nutrient analysis would be a good idea to just get a handle on what some of the levels are looking like and if any fertilization may be necessary or warranted. Okay, so what does the future look like with beech leaf disease? So we can look into our crystal ball. We've kind of gone through this already with beech bark disease. So we have the killing front where the disease is kind of sweeping through, resulting in widespread mortality. And that's the point we're in right now as it makes its way through New England. The aftermath forest is this some kind of new normal where the forests adjust to this major disturbance. And northern hardwood forests have already done this with beech bark disease. So they're going to have to do this once again after beech leaf disease moves through and we see this widespread mortality. So what's this aftermath forest going to look like? So again, we go back to the origins. The folks in the Midwest have been thinking about this as well. So this was an interesting study that came out looking at the distribution of beech leaf disease and beech bark disease in forests surrounding Lake Erie, which is where the epicenter was, and looking at future implications. And they noted there was an increase in sugar maple. That's good. We'll see an increase in sugar maple in our forests in Massachusetts and New England. There's a good seed source, a lot of mature trees. Red maple. We are so reliant on red maple right now in a lot of our managed landscapes and also our forests. The numbers of red maple just continue to increase over the decades as a result, excuse me, of chestnut blight, of spongy moth outbreaks. There's more red maple than there's ever been. It demonstrates a wide degree of plasticity. It can tolerate all kinds of different environments, and we really need red maple going forward. It's like the backbone of our forests, in my opinion. They found a lot of white ash. Okay, if this was a game show, this would be the big because of emerald ash borer. Ash doesn't have a future because of emerald ash borer. And then they found a lot of beech. So there's going to be a lot of root sprouts. There's also a seed bank, but another X and the wrong sound because it's just going to be continue to be infested and die back. So there's really no future for beech unless something changes dramatically. Now, if we look at our region locally, we could see an increase in red oak. We could see an increase in white pine, eastern hemlock, black birch is a species that tolerates some shade. So those are 4 species that I'll really look for, hopefully filling in. And that's unfortunately on the southern edge of the range of the northern hardwoods. You know, as you get, you know, for many of you that know, if you get up into northern New England, the northern hardwood forest, I mean, it's beech, birch, maple. I mean, it gets pretty restricted, and then you start to see red spruce and some other conifers, but you start to see the diversity really decline. It's, you know, for us here in southern New England, where you mix with the central hardwoods, we've got a chance to see some other species move in, but in a lot of the northern hardwood area, there's very limited diversity. We also have to deal with this problem of overbrowsing, which has it's been a major problem. It continues to be a problem. And, you know, how could we blame little Bambi over here? Look at how cute she's looking there. But there's been a number of studies that have shown just how destructive these deer populations that are way above their carrying capacity due to the loss of large predators, how damaging they can be. This study here, overbrowsing in Pennsylvania, in the title here, I wish I could use depauperate more in my like to add that to my lexicon and use that in day-to-day life. But this 60-year-old deer exclosure study, there's been a number of studies that you set up fencing to keep deer out. What's the forest look like inside there versus outside the fence where you have this kind of unchecked deer browsing. Browsing caused a 55 to 100% decline in black cherry, sugar maple, black birch, and alternate leaf dogwood. Beech is non-preferred browse for deer, unfortunately. So deer they will eat beech, but if there's a sugar maple, if there's a tasty little red maple seedling, they will gladly go after those. So they found a dominance of striped maple, Acer pennsylvanicum, and American beech, 82% outside versus inside the exclosures. So that's also a dose of bad news. There's areas around the Quabbin right now that are just devoid of trees in the understory. And you have these areas where hay-scented fern has kind of taken over like an invasive plant. In some areas, it's been a big problem. There's been controlled hunts around the Quabbin to bring the deer population down. But there's a real lack of regeneration in some forests where the deer population is so high. And to a lesser extent, moose populations. Moose can eat like 10 times as much as deer. And while they're not nearly on the same level of damage, they certainly can do their part at times. Okay, running out of time. So very quickly here. So this really points towards our need for diversity going forward. This little schematic on the left, say we have a disease of red maple. We have this small little susceptible red maple illustrated by this red square here. It attacks this red maple and kills it. It needs to find another red maple. Well, it has to move through this diverse array of other trees that it can't attack until it finds another red maple. And this is a large, mature red maple with good defense response, and it's able to fight off this disease. And so we don't see the disease continue on. But when we have monocultures, or when we have forests like our northern hardwood systems with all this understory beech, same size, same age, roughly same level of susceptibility, you get the disease establishing, and you get this increase in inoculum. Inoculum is diseased tissue that's harboring the pathogen or pest and allows it to spread. You get this widespread outbreak. And we have seen this before. We have, unfortunately, overplanting of American elm because it was such a successful tree in our urban environments. Dutch elm disease established, wipes out American elm. And we see that legacy continuing today with these high populations of Norway maple. Tree wardens were told at the time to replant with Norway maple. It's a great tree for our urban environments. And so this is why many of our municipalities now have such high populations of Norway maple. This is from the Boston Urban Forestry Plan, looking at the 10 most common species in certain neighborhoods. And almost 1/3 of all the trees in West Roxbury are Norway maples. As a result, it's a direct legacy of Dutch elm disease. Even when Norway maple is not the most abundant tree, we see other trees like honey locust. Honey locust is a great tree, certainly has a place in our urban forests going forward. It's native to North America, but we don't want 40% of it in any particular location. We want to see more diversity. If you happen to read Arborist News, if you're part of the International Society of Arboriculture, or even if you're not, this is a really good publication. My colleague Rick Harper and his graduate student put out this publication recently, Understanding the Relationship Between Tree Supply and Diversity. We have all these grand plans for diversity, not only in our urban environments, but in natural environments as well, in reforestation efforts. And unfortunately, those trees are simply not available to us from nurseries. If you go through, I was looking at Weston and Bigelow, what they've got on tap for the season. There's a lot of trees that we would like to plant that simply aren't available because it doesn't make sense for the nursery to carry them and sell them because the market isn't there. And so that's a big challenge for us going forward. Fagus crenata is a tree that seems to exhibit high degree of resistance to beech leaf disease. So this is a species that I'm sure will be looked at going forward as an ornamental here in North America. And I'm quickly running out of time here, so I'm just gonna very quickly go through this. Sorry for going over. This is just a European beech in Worcester that we're trying to protect and save. It's benefiting from isolation. It has some pretty serious decay issues right now. In the lower trunk, it's had a big tear out, and it has an infection of Ganoderma applanatum. This is a very common wood rotting pathogen of deciduous hardwoods. So this, again, points to just all the regular diseases that beech encounter. What we're looking at here are sonic tomograms. So this is some work I do in the lab for tree preservation, looking at the decay and the spread of decay within the trunk of the tree. So using sound waves, we can estimate the density of the wood, and we look over this this 4-year time period of 5 years, actually from 2020 to 2024. We see this green, purple, and blue area expanding, indicating that the decay is expanding. Here's another European beech with Ganoderma. Sorry, I've got to rush through these. I don't want to hold you up for too long. Again, some significant decay here. So this is a big hurdle in protecting some of these large and old trees going forward, is just the rigors of decay that set in for a lot of these trees. And here's a tree at the Ropes Garden under the management of Peabody Essex in Salem that I'll be working on this summer. And there's a lot of interest and effort to save these individual trees because of their cultural significance. So large swaths in New England dominated by beech saplings and pole-sized trees. Beech bark disease has set the stage for beech leaf disease and created conditions more conducive for an outbreak prior than forests would have been in prior to beech bark disease. We have very little chemical options, and some of the chemicals that do show efficacy are not ones that we want out in the landscape due to their potential persistence. And our aftermath forest should contain higher percentages of sugar and red maple, and hopefully some of the other associates as well. And this is my last slide here, just another one bites the dust of trees that we've lost from introduced disease: chestnut blight, Dutch elm, butternut canker, dogwood anthracnose, emerald ash borer. We could put spongy moth in here. It's resulted in widespread mortality, but not the loss of any particular species the way these have. But we will see how beech handles this demise over the decades to come. So happy to go to the subsequent breakout room for any questions that you may have, and thanks for your attention.
Awesome. Nick, thank you for a fabulous talk. Although I'm sure we you know we're waiting for the sunshine to get away from this seasonal depression, especially with this topic.
It's not a cheerful subject, is it?
Exactly.